When it comes to moving molecules across membranes, there's a lot of terminology which we'll review. But the most helpful thing is really just to logic your way through. The key things to keep in mind are whether the molecule is moving in a favorable direction. So whether it's kind of going the way it wants to go, thanks to its electrochemical gradient, and whether the molecule actually can go that way. So does it need help to get through the membrane? And if it needs help to get through the membrane, what type of help does it need? And that's going to dictate kind of how we classify the transports. Molecules, they want to move along their electrochemical gradient. That's their motivation. They move from where their concentration is high to where their concentration is low, thanks to just simple diffusion and random movement. And they're going to move towards a charge that is opposite to them, if applicable. And so we have this combination of our chemical and electrical gradients, our electrochemical gradients. If a molecule is moving in a direction that's along its electrochemical gradient, we say that's energetically favorable, and it's not going to require energy. In fact, we'll get energy. But if the molecule is moving in a direction that's against its electrochemical gradient, well, there we're going to actually have to put in some energy in order to get the molecule across. So even if we think, though, of a case where the molecule is moving along its gradient, so we'd have passive transport, it doesn't need like energy to go through, it still needs help to go through often. And so here is where we can talk about like facilitated diffusion, where we have a channel that's actually going to help let the molecule through. It's going to do things like offer a favorable passage, have a like hydrophilic pore in the center of this protein, so that this molecule, if it's not hydrophobic and it would be like scared off in the inside of this membrane, well, now you have this hydrophilic passage through. So that would be facilitated diffusion. It's going down its electrochemical gradient, but it needs help to do so. Only small, really small, like non-polar things or really small polar things like water, but water can also go through aquaporin, but very small molecules can kind of simply diffuse through the membrane. But if we're talking about anything charged, we're talking about an ion, we're talking about anything superpolar, those are going to need some help. Those are going to need your facilitated diffusion. So it's going along its gradient, but it still needs help. So first question, is it going to go along its gradient? If it goes along its gradient, it's going to be passive transport because you're not putting in energy. If it doesn't go along its gradient, it's going to need active transport. If it's going along its gradient, so we say it is going to be passive transport, you could still have facilitated diffusion where it needs some sort of channel to help it, or it could just be a passive diffusion where it can just freely diffuse through. One of the additional benefits of facilitated diffusion is that you can actually control the channels. So with voltage or with some ligand binding, this way you're actually able to keep that gradient going because, well, if things just diffuse freely in and out and you weren't taking things away or anything, what would happen is you just reach an equilibrium where your gradient would be gone and there would be no desire to move anymore. So those channels can be regulated, and those channels can therefore maintain these gradients, are able to prevent letting things in until you want them. And so this can be really good for sending nerve impulses and things like that, or quick signals into your cell. What about molecules, however, that are moving against their gradient? If we're moving a molecule against its gradient, this is going to require active transport. It's going to require the investment of energy. Often this energy is going to be coming from ATP, either directly from hydrolysis or through like phosphorylation, either of which can cause changes in the receptor shape, so conformational changes that might open the passage for that molecule. We can classify active transport as primary or secondary. And that depends on whether the energy is kind of being spent directly in the place that the molecule is going through or indirectly, in that it's being spent at a different pump that's pumping out a different molecule so that you're creating a gradient that this other molecule can like piggyback on. So because we can have co-transport, so we can have the movement of multiple molecules through the same transporter, either symport where they're in the same direction or antiport where they're going in opposite directions. This allows us to use a gradient of one to power the gradient of another. And that would be an example of secondary active transport when you're pumping out one molecule through primary transport, but then using that gradient of that molecule to bring in the second molecule through the secondary active transport. So the names get a little confusing, but the difference is whether you're spending that directly at the receptor, at the transporter that the molecule you're thinking about is coming through, or you're spending it on some other transporter for a different molecule, who that, in order to create a gradient of that molecule, which you can use to power the import of your molecule of interest. Decision tree time. You want to consider, is the transport against the gradient, in which case it'd be active transport, or along the gradient, in which case it would be passive transport. If passive transport, can the molecule freely diffuse through the membrane? In that case, it'd be simple diffusion. But if the molecule can't get easily through, well, now it's going to need some help in the form of an integral membrane protein. This protein is going to offer a channel that provides a more favorable passage, such as having a hydrophilic pore that these molecules can move through. We can think about controlling that pore, like gating the channel. The channel might be gated by something like charge, like something like a voltage gated channel, which is common in things like neurons, or it might be regulated by like a ligand binding, so a ligand binding channel. Those are all still going to be examples of passive transport because you're moving along your gradients. However, if you want to move up against a gradient, well, now you're going to have active transport. If you have active transport, your options are going to be primary or secondary. In the case of primary, you're spending energy directly at the transporter. And so this might be through hydrolyzing ATP, such as in the case of our ABC transporters, or through phosphorylating the protein in the case of like our P-type transporters. And in those cases, you're spending the energy directly at the receptor in order to cause a conformational change in the receptor, so that you're able to, say, open it up for the molecule to go through. In the case of secondary active transport, you're using the favorable transport of one molecule along its gradient to power the unfavorable transport of the molecule you're interested in against its gradient. And so you still have to spend money, but you're spending money in a different way in order to have a gradient that you can use to power the movement of this thing coming through. Because when you go along your gradient, you're going to release energy, but if you go against your gradient, you're going to require energy. So the movement of one molecule along its gradient can provide energy to power the movement of another molecule against its gradient. And that is secondary active transport. So your secondary active transporters are always going to be co-transporters. They're going to be transporting multiple things, but other types of transporters can either be uniports, they're transporting a single thing, or they can be co-transporters. And then for each of the molecules that are going through, you have to think about, is it going along its gradient or against its gradient? And if they're going along the gradient, then great, it's going to be passive. If they're going against the gradient, you're going to need some energy. And if one's going in or against and one's going towards, it's probably an example of a secondary active transporter.